Okay, welcome. Today I will start talking about biochemistry. We will talk about proteins. Proteins, it's one of major biopolymers. Originally, it's mean bio is mean alive, polymers it's mean same fragments repeat many times. For example, if we have any substance where each element repeat, it's monomers. Poly, many, mers, fragments. So biopolymers, it's polymers where same fragments repeat many times. And if we're talking about most important biomolecules, polypeptides, nucleic acid and sugars, polypeptide and nucleic acid, and particularly sugars, not all sugars, it's polymers. And most important, most valuable polymers for a live creature, it's proteins or polypeptides, depending classification. Proteins consist of amino acids and form giant chain. Here show some amino acids. If we're talking about general structure, it's consists of COOH, it's acid, NH2, it's amino group, and some radicals. So amino acids, it's acid, it's compounds with acidic and amino groups. And here I show you some of them. All amino acids we can group by 1, 2, 3, 4 groups. It's green, shown by green color. It's amino acid where radicals non-polar. It's alanine, valine, isoleucine, leucine, methionine, phenylalanine, and many more. Another groups, it's with polar side. In other groups, it's acidic amino acid and basic amino acid, and special case. So in general, we have about 21 amino acids, and all amino acids we can divide by two groups. It's essential and non-essential. Non-essential, it's amino acid which can be synthesized from essential. And essential, it's unreplaceable. For example, some food additives for growing animals in farm. Food additives have highest concentration of essential amino acid, because if without essential amino acid, non-essential without essential amino acids, another amino acids can be replaced. It's like, for example, if you produce car and you haven't enough wheel, you can produce car. Even if you have a lot of engines and a lot of frames, you can't replace wheels by something else. And amino acids, because they have acidic and basic amino groups, they exist in very strange form, on example with glycine. As you probably know, if you have, for example, ammonia and add, for example, acetic acid, you will get salt. Acid react with base and forming salt. I see, and I show it before. We get C3, C, O, O, O minus, NH4. We have cations and anions. Same situation we have with amino acid, but with difference. In amino acid, we have amino groups and we have carboxylic groups. And one fragments of molecules react with another fragments of molecules, and we get NH3N plus OO minus and R. And these anions we name as Zwitterions. It's charged, it's salt, and most amino acid in pure state exist in Zwitterions form. But it's salt. It's salt of amino acid with itself. And Zwitterions, it's very important. These molecules charged, but because overall charge 0, these molecules can be moved under voltage. So in electrophoresis, this Zwitterion not moving. If we have several amino acids, we can get peptide. For example, we have amino acid NH2C. For example, glycine. And this glycine react, for example, with, let's react with, for example, alanine. Oh. If wait, if amino groups of not all, if amino groups of valine react with acidic group of glycine, we can get new compounds, and we name it peptide, and in result we will get CH. We have peptide, and for dipeptide, it be more specific. And for these peptides, we have several ways of coding this. We can draw chemical formula, but it's too big for drawing, especially if you have giant chain. We have 3-letter codes. In our case, we have our glycine. You see, glycine. So glycine and alanine, it's alanine. It's important. It's not alanine glycine. It's glycine alanine, because in left side we will show N chain. It's part of molecules which have 3 amino groups, and in right side we have C end, acidic end. But even this 3 letters code, it's quite complicated, because if you're dealing with, for example, with several hundreds of amino acids, it's very hard to use this long code. And for amino acid, they have 1 letter code also. For glycine, it's G. For alanine, it's A. So simplest, another way of coding these molecules, it's GA. So polypeptide we can represent by 3 ways. At first, we can draw formula itself. We can draw 3 letters code, and we can use 1 letter code. All 3 cases meaning same. It's peptide. In our case, it's dipeptide. Some of proteins were important. For example, here some examples. Hemoglobin, it's compounds which transfer oxygen in our blood. Insulin, it's compounds which regulated glucose exchange, energy exchange. Oxytocin, it's also polypeptides. It's hormone of happiness, and collagen, it's need our body as mechanical connections. Polymers made from amino acids, name polypeptides or proteins. And these proteins can be differently organized in space. Sequence of amino acids, as in our case, for example, glycine, alanine, valine, glycine, alanine. It's primary structure. It's just sequence of connected molecules. In space, these molecules can be organized differently. We can group in this chain. For example, we can group in it in helix or sheets. In this case, we're dealing with secondary structure. And even this helix layers or just lines can be grouping in more big fragments and forming something like tertiary structure. It's when molecules are organized even in more complicated structure, and all proteins sometimes it's a tetrahedral structure if several proteins combine together. And one of example it's insulin. Insulin have several fragments of polypeptides, and all these fragments polypeptides it's separate chain. So what you must memorize? What most important? Amino acid sorry. Amino acid, it's polymers which consist of acidic group, amino groups, and some radicals. Amino acids can form polymers, and these polymers we name proteins or peptides. For peptides, we have several ways of coding. We can use 3 letters code, we can use 2 letters code. 1 letter code, sorry. And we have several type of organizations, this structure in space, primary, secondary, and tertiary. For today, I think it's all what I planning to say. Don't forget, I also have TikTok. In my Facebook group, I can show you link. It's actually very interesting fragments in formats of explain. Just who not watching it before, I will show you. Wait a second. For example, it's how acids react. Quality not very well, but if you're interested, please watch. It's more short way than my YouTube video. So for today it's all. Thank you for your attention. Subscribe, share, and good luck in preparation.